And finally, in the last section of this paper, we're talking about mutations, the use of mutations in genetic analysis. Again, I've mentioned this in class. Remember, I talked about the bread mold, the Neurospora crassa. I talked about I talked about minimal medium. You expose the Neurospora crassa with what, maybe radiation, and then you grow it in rich medium so everything can grow, and from whatever grows in the rich medium you transfer it to minimal medium and see what doesn't grow, right? So then if you find something that grows in rich medium but doesn't grow in minimal medium, then you take that particular and then you test it with different vitamins or amino acids, say for example you give it histidine, it grows, and then so now you have a mutant that is unable to make histidine, so it's a histidine mutant. I mentioned this also with relevance to Jurassic Park. If you remember why I did not concern that these dinosaurs do not leave the island and perhaps invade other regions, is because they said these dinosaurs are dependent on all the plants and animals in the island have been provided with I think a particular amino acid, I think histidine, I think, and all the dinosaurs cannot live without this supplement. So that's why I mentioned here, and that's why it's actually explained in some sections in this genetic analysis. So talking about because what's important in the classical example of genetic analysis, we study mutants. We don't see how a normal gene functions. When we study is that you want to study a normal gene, you knock it out, you make a mutation so that it doesn't function, and then see what happens to the organism. So if this gene you do crosses so that this gene is mutated, and then you'll find that the organism keeps falling down, so that the gene is important in balance, maintaining balance, so that kind of thing. So here it says genetic screens exposing a population of organisms to physical mutagens. You create mutations and then you see what the effects, and then try to repair the effects. That's how you actually identify mutations. So this is again about genetic screens. The concepts of prototrophs and auxotrophs. Prototrophs, again, if you know, there's another here you go. A bacterial cell with the wild type metabolic properties of species is a prototroph. That means it can do anything, so that means it can live in minimal medium. Whereas auxotroph are mutants that requires supplements to live. So well, you read that this one is conditional mutants. That means it says that if you want to study replication mutants, you cannot, because if it's mutated, then of course it's going to die. So how do you study replication if you cannot mutate things? So there are some systems where it can live in certain conditions, but like for example, it can live in lower temperatures, but it cannot live in higher temperature. Right? So these are what you call as conditional mutants. Right? So, and this have given rise to the ability to study some proteins in different conditions. All right. So, okay, I will not go through further. So again, remember some of the terms that have used in this, and some of them are the subject of the quiz. What they are and so on. All right. Okay. Good luck.